So we're going to do a quick review because we're going over the 20 amino acids. Naturally, I should say 20 naturally occurring amino acids. These are the ones that are in the. So that's my order. These are the ones that are used to make proteins in vivo. Right. That's not to say that they're the only amino acids, because there's some others that you actually learn about next semester. For example, like ornithine, citrulline, things like that. But they just are not used to synthesize proteins. But they too are important. And there are some that are not alpha amino acids as well. But we were going through and we went through the ones that were nonpolar. We had leucine, valine, alanine, glycine, which isn't on here, proline. Remember, proline was special because it's the only one that doubles back on itself to where it makes 2 points of contact with what's considered the backbone of the amino acid. And methionine. And then here they're showing phenylalanine, which is also an aromatic one since it's got the benzene ring. Isoleucine and tryptophan, which is also aromatic. Then we talked about those with the polar side chains. We went over asparagine and glutamine. Those are the two amides. Serine, which is the alcohol, and threonine, which is an alcohol, but it's off of the first carbon, so it's branched. And then this was the space-filling molecules. And so this one has the glycine in it, serine, so on and so forth. And just like asparagine and glutamine are related in the sense that they're the same, except there's an extra methylene group between the two. That's what it is with aspartate and glutamate. Which technically, the way that this is shown, those are aspartate, glutamate, and those is aspartic acid and glutamic acid. I couldn't go through and change that because that's the way that they made it. Okay, and then we talked about cysteine. In fact, cysteine can form disulfide bonds, and then tyrosine. And tyrosine, cysteine, and serine are the best three at being nucleophiles and doing catalysis, typically. Threonine to a lesser extent, and the carboxylic acids also to a lesser extent. Okay. So, we have that glutamate and aspartate have an extra carboxylic acid. They are very, very important in doing what's called general acid-general base catalysis. So if you've already started working on your enzyme topics, then you may come across that in the mechanism. Okay, and so many times things like glutamate, aspartate, histidine, which we haven't discussed yet, are very important in doing that because they can be both hydrogen bond donors and acceptors. Okay? But if this was a free amino acid floating solution, it's going to be negatively charged at neutral pH. Okay? So at pH based off of your body, it's going to be negatively charged because that terminal carboxylic acid has also lost its hydrogen. Now we have arginine, lysine, and histidine. Okay? So, arginine, if we go back to, let me change the color here, it's easier to see. Just to the backbone, remember, all amino acids have the same backbone, except for that proline's nitrogen is also connected, but we've already done proline. So this is what it looks like, and it's just this R group that's hanging off that's changed between them. Arginine is a little tricky. It's basic, as in it's a base, not as in just basic as in simple. It has 1 carbon, 2 carbons, 3 carbons. Then it's a guanidino group. Yes. Okay, I've shown it in its charged form. When I remember there was 3 linking carbons, and it goes NCN, and then that middle carbon here is double bonded to what would be like an imine. That's why the number is. But what happens here is that there's resonance between the 2 terminal nitrogens. So there's actually resonance that occurs between these 2. I've just shown the positive charge being on one, but it's not like there was one is more special than the other. So this is a base, it accepts hydrogens easily. In fact, you have to go to really high pHs in order for it to lose that extra terminal hydrogen. So that's why arginines typically have a positive charge overall at neutral pH. Okay. Arginine, what's its 3-letter code? Yep, it's ARG. And its 1-letter code can't be A because A was, or is, alanine, and so it's R, which actually is at least phonetic, arginine. So the hardest thing with arginine is just drawing it. It and lysine are both very, very long. Okay. So, lysine is very similar. It too is a base, and it can be used as a nucleophile. In particular, it's used in something called a Schiff base, where it can form reversible imines in reactions. So it has the longest chain of just carbons in a row. So it has 1, 2, 3, 4, and the 5th position is an amino group. And I'm showing it charged because under neutral pH, that terminal, 1, 2, 3, 4, yep, that terminal nitrogen would be positively charged. And that's lysine. Not glycine, but lysine. Remember, glycine is spelled with a C, lysine is spelled with an S. And whenever I say imine, what I'm saying is that what makes this one special, I'm not going to redraw the entire side chain, but this was a side chain. That terminal nitrogen can form reversible double bonds with carbons. And so then water can come in and knock it off to where you get the free lysine on the enzyme back and you get an aldehyde or ketone on the other end. So it's one way that you can actually swap and trade groups, transfer things from one to another. We're going to see it all the time in one specific class of enzymes called aminotransferases, which are very important next semester. But some of you may even be looking at aminotransferases as your enzymes, or a specific type of aminotransferases enzyme. And almost always, they use the enzyme would use a lysine's nitrogen to do that, because it can form a temporary Schiff base, which is just another way of saying an imine. And then water can come in and knock the nitrogen off to reform the aldehyde or ketone, or carboxylic acid. Just anything that has that carbonyl group. Okay. So what's lysine's 3-letter code? It's really easy. LYS. It makes sense. Its 1-letter code is not L because L is leucine. It's not Y because Y is tyrosine, and S is serine, I isoleucine, N asparagine, and E glutamic acid or glutamate. Because it's the one that spells glue whenever you put the 3-letter and 1-letter code together. So that means it has to have a different letter. And so, does anyone know the letter for lysine? It's capital K. Yep. It's one that people tend to get wrong. But just like tryptophan can get people mixed up once in a while. Lysine's another one. This is kind of random. Next up is one of my personal favorites because you're going to see it a lot, and not only in biochemistry but in neurology, neurochemistry, because it's related to neurotransmitters and it's very important. That's histidine, because it too is a base, and it's involved in a lot of general acid, general base chemistry. It has a linking carbon, and then, whoops, it did not come out very well. Whoops. Let me just see if I can draw it a little prettier. Linking carbon, and then hang out that carbon. I draw it like home plate once again, but there are two nitrogens and another carbon down there. And then there are double bonds. There's a double bond to the top, and it doesn't matter which of the nitrogens you put as a double bond. Okay, this is in its unionized form because its pKa is right around neutral pH. And see, there's already a hydrogen hanging off of this nitrogen. Many times it's just implied, but there's a hydrogen there. This other nitrogen can also be protonated. It's the one that has the pKa. So sometimes it can look like this, whichever one you put the double bond. The reason why it doesn't matter which one you put the double bond in is because this is aromatic, so there's resonance actually between the two nitrogens. That double bond can go back and forth, which is one of the reasons why it's really good for general acid, general base chemistry. And when we do the enzyme chapter, I'll actually show you this in at least once or twice, because it can be attacked on one side. Let me erase it. It can get attacked here, and then the double bond just flips over to the other nitrogen, and then this hydrogen can actually then go someplace else with electrons. So that's why it's sort of like little bumper cars. One nitrogen gets attacked, the double bond flips over, and then the hydrogen that was attached to that can go to something else. And then it can be reversed, because then something can come and grab the hydrogen, double bond flips back, and so on and so forth. But we'll see that time and time again. So let me erase that part now. Okay. And what's histidine's three-letter code? HIS. It's his, and its one-letter code is H. Okay. And so here's just some of the ways that they show it, except you just have to, that is not a too negative charge on that histidine or on the arginine or the lysine for that matter. That's just showing you where the point of attachment of the R group would be. Then we have the space filling diagrams for them. You can see how long lysine and arginine are compared to some of the others. Okay, and then in the PowerPoint, I did include the one-letter and three-letter codes for you. Okay, please note that some letters are missing. For example, X is missing. There's not an X. There's not a Z. Okay. And some of these, it's because there are times that there are certain, and there's not O, because technically O would be ornithine. There are times when you can't tell if it was asparagine or aspartic acid due to degradation, and so then they use some of the other letters like B or X to either say it's asparagine or aspartic acid, or that was glutamine or glutamic acid. Because if you remember from Organic Chem 2, that carbon, the carbonyl group, can get attacked and water can leave, or ammonia can leave, so you really couldn't tell if you really started off with a carboxylic acid or an amide. So that's why they have a couple of letters that are, if you don't know which of the two it is. I wouldn't expect you to know those letters. I would give you that in a key. Okay, so all 20 are alpha amino acids except for glycine. All of the alpha carbons is a stereocenter, so they're all chiral except for glycine. Isoleucine and threonine actually have 2 chiral carbons, so that makes them special. We've already talked about how proline is special because it's fixed, it's rigid. Then we have some that are the uncommon, which really are just amino acids that have been post-modified. Sometimes these are called post-translational modifications, because after it's been translated into the protein, then they get modified to do special things. So one of the most highly modified ones is actually proline. Okay, and so what happens is we have proline that gets hydroxylated, so it gets an alcohol added on to one of the carbons, and it makes hydroxyproline, which, why would that be important? What can that proline now do that the original proline could not do? Hydrogen bond. And we're going to find out it's very, very specific why that's important. Likewise, we have hydroxylysine, where it too can hydrogen bond now, because it has this alcohol group that's hanging off of the end. Then you get thyroxine from tyrosine, which is highly iodinylated. So it has a lot of iodines on it. Okay, so that's also important. These are just some of the examples. Okay. So the reason why, oops, let me go back. Hydroxylysine and hydroxyproline are important. In fact, in collagen, I do believe usually for most types of collagen, it's right around a third, right around 30% or so of the amino acids are hydroxylated prolines or lysines. Okay? Why does that make sense? What is collagen? I mean, yes, it's connective tissue, it's given to you, but what does it mean? What does it do? What's the whole purpose of collagen? Keeps it strong. Where do you have a lot of collagen at? Your skin. So what happens as collagen starts to break down? You do get wrinkles. There's actually a disease that's associated with the hydroxylation of prolines. And it's where, I can't remember the official name for it, but sometimes it's called skin slippage. And so one of my friends, who worked at the PTO, when she first started there at the Patent Trademark office, she actually had to share an office with the guy. Like, they had a little office, but it's 2 people at the office, and he had that disease. And so what happens is, like, his skin really would kind of sag, or it's almost like an overripe tomato. Okay, but what happens with collagen? So what does it look like? Does anyone know what collagen looks like? It's not a sheet. It's actually much more stronger than a sheet. It's fibers, collagen fibers. And so it's a lot like a rope. When we talk about alpha helices, it has a special type of helix. And then there's like 3 of them that get wound together. I can't give it justice because I can draw DNA double helices. But adding a 3rd one in makes it a little more, it's not too shabby. Okay, but there are 3 helices that all get wrapped around together, kind of make like a rope. And what happens is you get these, first of all, the prolines help make it kinky, but then the prolines have alcohol groups that hang off, so then in between the little strands, they can hydrogen bond, it makes it even stronger. Okay, just like, the more strands around a rope, the stronger the rope is. And so then what happens is if you have this disease where you don't hydroxylate your prolines or your lysines, especially your prolines, enough, then you aren't making as many hydrogen bondings between the strands, and so then your collagen is not as strong as it should be. And thyroxine is found in the thyroid gland, which makes sense, consider that's where iodine is required and used. Then we have this all went backwards whenever it got updated to the more recent edition of Office. We have amino acids and neurotransmitters. Okay, so we have serotonin, which what is serotonin? It's a neurotransmitter, but what's some of the things that it regulates? we're going to talk about a lot next semester. But serotonin can do some with sleep. What else is it? That's more like a lot of people say, oh, tryptophan. It's been linked to sexuality. It's been linked to moods, depression versus, moments of elation. I mean, it's a major homeostatic control. Oops. I have to do something. I'm not going to give it away because it's tryptophan that's converted to serotonin. We actually go through that metabolic pathway next semester, since almost all the next semester it's really just metabolic pathways. We have the very first chapter, which is on bioenergetics, since it's important, and then the rest of it's pretty much just metabolic pathways. Okay. Oh, come on now. Okay. So body temperature, mood, sleep, appetite. Okay. All right. And so you may have heard of SSRIs, which are selective serotonin reuptake inhibitors. In fact, a lot of times these drugs are they counteract with other drugs. And so it may say, don't take such and such if you're taking either an MAOI, which is something else, an SSRI or something like that. And it's because these antidepressants, what they do is they block the ability of serotonin to be taken into the nervous system, and so therefore you have more serotonin floating around in your serum. So therefore, if it stays around longer, the idea is it's going to keep you from being depressed. Of course, many of these also have can have very bad side effects, since serotonin is used in many different body processes. And then, you gotta be kidding me. Tyrosine is used to make epinephrine and norepinephrine, for that matter, which is the same thing as adrenaline and noradrenaline. Dopamine, those are all tyrosine based. And like I say, next semester we go over the enzymes that are used to make that, and how you go from tyrosine. What about, so how many people take antihistamines? I take antihistamines. Like what's an example of antihistamine? What's a common antihistamine that people can take? Pardon? Claritin, Benadryl, those kind of things. All right, but what are antihistamines? They help with your allergies, but what are they going against? Histamines. And where does histamine come from? Pardon? But how do you... Sorry. I left my normal status at home, and this is like a freebie that I picked up at a conference, and it's like coming apart in my hand. Where does it come from? How did the cells even make it? Make histamine. Histidine actually is decarboxylated and it becomes histamine. Okay, and so once again, we will go over that kind of stuff next semester. Okay, now one thing I do want you to know is the ionization of amino acids. So when it's in their Zwitter ionic form, that's whenever the amino group has a positive charge, the carboxylic acid has a negative charge, it's net neutral. If you go to a low enough pH, you can actually force the carboxylic acid to take that hydrogen back. And so now it has a net charge of positive 1. If you go to a high enough pH, not only does the carboxylic acid lose its hydrogen, but so does the amino group. And so now it has a net charge of negative 1. That's true for all of the ones that don't have any charges on their R group. Okay? So for every titratable hydrogen, you're going to have a pKa. All of them will have at least 2. Okay? Now, one thing I want you to know is please don't memorize. I'll either give you a table, or you should know the relative numbers. So what I'm saying is, for example, all of the ones around the carboxylic acid for this hydrogen right here usually are between 2 and 3. The pKa is between 2 and 3 pH. Just like all the ones for that nitrogen, for this one to fall off, to become neutral, they all run usually between 9 and 10. There's a couple that go beyond 10, but just know the ballpark figure unless I give you the specific number. And I would always give you the number for the side groups, side chains, like for glutamic acid, aspartic acid, histidine, serine, tyrosine, so on and so forth. Those that have the hydrogen that can pop on and off from their R groups. But it's important that you know what that means, because we're going to be doing titration curves just from like Gen Chem, and you can figure out what the net charge for a protein or a peptide is based off of the pKa's of its amino acids. And I'll show you what I mean by that. So the average pKa of that carboxylate, or carboxylic acid to become a carboxylate, is 2.19. Once again, just remember it's 2 to 3, which is much stronger acid than acetic acid, which for those who haven't had organic chemistry yet, or if you've just forgotten, acetic acid looks like this. Of course, there's a CH3. So acetic acid, this hydrogen pops off around 4.7, its pKa is 4.76. So that's why they like to compare acetic acid to the amino acids, since all of the amino acids are alpha amino acids. So the R group is hanging off of that C where the H3 is. So why is it that, it may not sound like a whole lot, being 2.5 pH units away, but remember this is a base 10, so that's more than 100-fold stronger acid than acetic acid. So what is it about an amino acid, when we draw it, remember it looks like this. This is in its unionized form. So what is it about this amino acid that makes the H pop off so much more easily? Because they both still would have the same amount of resonance, so the conjugate base would still both be a carboxylate, but there must be something else nearby that helps perturb the pKa of that amino acid, carboxylic acid. It's the nitrogen. Okay. Oops, there we go. All right. It's due to the fact that nitrogen there, remember that whole idea of inductive effect from organic chemistry? In fact, we just started covering it this last class period or so, this last week in organic chemistry, if you're currently in there. That inductive effect, the ability of the nitrogen is close enough nearby that it can help, I hate saying pull the proton, because it's actually, the electron density is left behind on the oxygen, but it can attract that proton because it's a base. And so since it's a base near an acid, it's going to pull the hydrogen off. Okay. And so that's the reason why it's much more acidic than what you would expect, and it has, under physiological pH, it would have, if it's a free-floating amino acid, it would have its Zwitter ionic form. I keep saying free-floating amino acid because we're going to find out once you start building a protein, you're actually going to get rid of the charges on the nitrogen and the carboxylic acid because they're going to be amides. So you're only going to have those charges on the two ends, on the N-terminus and the C-terminus. People forget that whenever they're figuring out many times whenever they're figuring out the charge on a protein. All right, I would expect you to know this. This is from Gen Chem. Many times we do it this way. Oh, come on now. All right, where you don't have to, but where you're adding hydroxide and then you're just measuring pH. Remember that was the titration curve. If you do it the opposite way, if you're adding acid, then it actually would look more like this. You know, it would be the opposite direction. Because you're starting at a higher pH and you're going to a lower pH. But usually we start at low pH and add to high pH. It should make sense at a low pH, so you haven't added any more hydroxide, that overall it's going to have a +1 charge. This is just for generic amino acids, say for like glycine or alanine. This is not going to be true for histidine. I mean, histidine is going to be positively charged at low pH, but you have to realize that it's going to have, instead of 2 pKa's, it's going to have 3. So for you to get it fully titrated, instead of going to 2 molar equivalents of hydroxide, you'd actually have to go to 3 molar equivalents because it's got a third hydrogen that gets pulled off. So that's why I mean that you'd have to, it's going to look a little different. For like histidine and serine and cysteine and some of those that have an extra hydrogen. Either they're an acid or a base. Alright, but if you start at low enough pH, it's going to have an overall positive charge because of the fact that the nitrogen has both the nitrogen and the carboxylic acid have their hydrogens. But as you start to add in hydroxide, it's going to first pull off the hydrogen from that carboxylic acid. So once you've added, let me erase this a little bit. Once you've added 1 full equivalent of hydroxide, that means it's pulled off 1 full equivalent of H plus. And so that's where we get, at this point, the amino acid would look like this is what our ionic form. So if you think back to calculus, you have this little inflection point. That's that first pKa. That's where the average, I think this one's actually glycine, is like a 2.34, but it's really between 2 and 3. That's the inflection point. It's the first pKa. Now if you keep on adding, and that pKa means it's 50% has a +1 charge and 50% is neutral. So overall, if you add 50% of the +1 and 50% of 0, what's the overall charge on that solution? It's going to be +1/2. Okay, so that's what the effective charge would be. Okay, now if we keep on adding, let me just switch colors. Now if we keep on adding a second equivalent of hydroxide, we're going to be able to, and you go to high enough where you added 2 full equivalents, you're going to pull off the extra hydrogen that had been on the amino group. So now it's got a net -1 charge. But once again, we have an inflection point where half of it has the neutral charge and half of it has a -1 charge. That's why overall it's going to have a -1/2 charge, the overall solution. And that's the second pKa. This is the reason why, as long as you know the pKa's, you can figure up the net charge on a peptide or a protein just by literally adding up all the various positive or negative charges. And there are examples that if we don't get a chance to go over them in class, I also put some in the suggested problems at the end of the book, or at the end of the chapter, I should say. Okay. So you have to make sure that you understand what these indicate and how to draw them. I can almost guarantee you a quiz or exam question will have something to do with this. So it's really important to understand what pKa really means. Overall, every amino acid will have what's called the isoelectric pH, which they give that pH as a special P number, and it's called a PI. And that's where the net charge is 0. And so for glycine, or the ones that are the simplest, where you only have 2 pKa's, it's easy, it's the average. So it's 2.35 plus 9.78, you add them together, you divide by 2, you get 6.6, and that's a PI. For those that have more complicated ones, then it's that, if we go back, it's the overall point here, the overall inflection. Whoa, whoops. I didn't have it on draw. It's this point where you have this net of 0. So, for example, with histidine, it's going to take 3 hydroxides to pull off all 3 hydrogens, it's going to be the position where you have 1.5 hydroxides added, 1.5 molar equivalents of hydroxide added. Okay, and so I would give you this table, or at least the table of the side chains. But you can see these are the simplest ones: alanine, asparagine, so on and so forth. They don't have measurable pKa's of their side chains. Serine does, but it's really high. That's the reason why you can pull it off. Or I shouldn't say it's really high. It's at a pH that's physiological possible, especially inside an enzyme, because within the environment of the enzyme, it makes it more easy to pull the hydrogen off for the serine to act like a nucleophile. Whereas those that are truly acidic and or basic side chains, then those I would definitely give you this. And so I would give you, these numbers right here. Now I would just expect that you can see that almost all of these are between 9 and 10, or roughly around 9 and 10. All of these are usually between 2 and 3, roughly. So if you were at pH 7, on histidine, you're going to, at pH 7, will that carboxylic acid have its hydrogen, or will it have already lost its hydrogen? It's lost it, because 2.05 is lower than 7. Okay? Whereas, will the amino group have its hydrogen, or will it have lost it at pH 7? It'll still have it. And then we have it at 8 o'clock. At 8 o'clock. It is too early this morning. At pH of 7, if it's got a pKa of 8, will most of it be, most of the hydrogens be on it, or will most of them be off? It's still going to be mainly on. It'll still mainly have its hydrogens, okay? It's just that there is a fraction, a percentage, one off is around 10% or so, that would actually be minus charge. But I wouldn't expect you to go to that, get it down to that precise. That's the reason why cysteine, it's actually easy to perturb the pKa just enough so it can lose its hydrogen to become a thiolate. And then overall, its pi is 5. Okay. And then we have the bases. So that's why, whereas histidine, its side chain is 6. So it can gain and lose that one really easily around physiological pHs. So, let's see. Yeah. Mm-hmm.
Does that mean like cysteine with the side chain being deionized before the amino group?
Yes, exactly. Mm-hmm. So what she's saying is, let's say that we go to pH 9. At pH 9, cysteine would look like this. It's going to still have that alpha. Well, it won't let me draw the hydrogen then. Did it come through? Barely. The alpha one, the carboxylic acid had lost its hydrogen a long time before. The nitrogen still has its hydrogen on, though, because it doesn't fall off until pH 10 or above. But the sulfur has lost its hydrogen already at pH 9. In fact, only around, 10% would still have a hydrogen. It would be primarily minus charge. So that's what that would look like. It would look like a thiolate. And one thing I want to make clear is that whenever proteins and peptides start to fold, it can perturb these pKa's because it can create an environment. That's why I keep saying this is if it's free floating in solution, but it can create the environment to where now that side chain's pKa is no longer at 8. Maybe it's now at 5. And so then if it's at 5, then it's going to always look like the thiolate under physiological conditions. And so that's the reason, that's how enzymes a lot of times work, is the way that they fold and everything. They create an environment to where it perturbs one or more of these pKa's, especially with serine. Because serine, if you saw their the pKa wasn't even listed because it's normally like at 10 or 11. I mean, it's really high up there. So normally you wouldn't think about it in physiological conditions. However, once it gets inside the protein, you can actually perturb it. Okay, that is possible. And that's why they do like pH profiles of enzymes a lot of times to see like what happens to the important pKa's inside the enzyme to make it physiological possible. Whoops. So let's just do an example of what if we had a tripeptide at pH 7. And we're going to say it's a tripeptide of, and it always goes from the N terminus to the C terminus. So it's going to be arginine, alanine, and we'll say cysteine. So RAC. So, and the question asks you, what's the overall charge of this peptide, assuming that you can use this table? You know that I mean, a tripeptide really can't fold very much to perturb anything. So where people make a mistake, let me change colors. Is remember this is the N terminus. That's not asparagine. That's the N terminus, and this is the C terminus. That's not cysteine. It's the C terminus. So if you want to put circles around them or something, you're making notes to where those are not the amino acids. At pH 7, what's the charge on the N terminus of the? Is it going to have the extra hydrogen, or has it lost it? Because remember, its pKa is usually around 9 or 10. It's going to have it, so that means there's a +1 charge on that one. Likewise, if we think about the C terminus at pH 7, does it have the hydrogen, or has it lost it to where it's negatively charged? It's lost it, so it's a -1 charge. Now we have to think about arginine. This is, let me see, this right here, that amino terminus, is the amino end of the arginine. So it's already been taken care of. Its C terminus doesn't exist because it's now the amide bond that's connected to the alanine. So people would forget and they say, oh, well, it's got a -1 charge hanging off of the other side, but it doesn't. And I'll show you what a peptide bond looks like. Because it's already bonded to the alanine's amino terminus. And so that's why the only part that matters is the side chain of arginine. So we have to think now, is the side chain of arginine, which is at 12, which has a pKa at 12, so at pH 7, is it going to have the extra hydrogen to be positively charged, or has it lost it? It's going to have it, so it's got a +1 charge. Alanine, both sides of the alanine on the backbone are in bonds, so they're not charged. So people always would forget, and they think, oh, there's a +1 on one end and a -1 on the other end, but neither one of those exists because those are both bonded to the neighboring amino acids. And so then we just have to think about the side chain. The side chain of alanine is just a methyl group. Methyl groups have no pKa. They have a pKa, but it's like 45. Something ridiculous. I don't even know that number. So it's neutral, so I'm just going to put a 0 there. Then we have the cysteine, and the cysteine, the N side of the cysteine is in the bond to the alanine. The C side we've already taken care of. That's the C side, the carboxylic acid side. It's the C terminus. So now we just have to think about cysteine's side chain, which is 8. So at pH 7, will it predominantly have its hydrogen, or has it lost it already to make a -1 charge? It'll have it. So it's also, the R group is also neutral. So now we just add this all up. We have 2 pluses and 1 minus, so overall this has got a +1 charge. So that would be the answer for this peptide at pH 7. Now if we went to pH 10, it'd be a different ballgame altogether. Okay, so, or if we went to pH 2, then you have to figure out, at pH 2, then that C terminus may have its hydrogen on it, or at pH 12, then the N terminus may have its hydrogen, had lost its hydrogen, as well as the cysteine has lost its extra hydrogen. So that's why you have to pay attention to where the pKa's are. And so I say just practice some of these to get the hang of it. Okay, and so that's where we're going to end for today, and we'll start back up with electrophoresis